Welcome, everyone, and thank you for tuning into this Genetic Engineering and Biotechnology News webinar. It's entitled Translating Next-Gen Sequencing from the Lab to the Clinic, Challenges and Solutions. Next-generation sequencing, more commonly known as next-gen sequencing, has revolutionized the study of DNA. Compared to the classic Sanger method, next-gen permits the sequencing of DNA and RNA more quickly and less expensively. The impact on basic research has been breathtaking, and now the technique has reached the point where it can begin to answer questions that arise in the medical clinic. To discuss this, we've assembled a very knowledgeable and expert panel. Let's meet them. Dr. David Gallus serves as principal scientist at the Pacific Northwest Diabetes Research Institute. David is going to discuss the technical advances in sequencing made by next-gen approaches and explore some of the challenges that remain to its successful implementation. Dr. David Smith, professor of laboratory medicine and pathology at the Mayo Clinic, will provide specific examples of next-gen sequencing in clinical practice at the Mayo Clinic. Dr. Elaine Martis is professor of genetics and co-director of the Genome Institute at Washington University School of Medicine in St. Louis. Elaine will focus on the application of next-gen sequencing in cancer genomics to guide therapeutic strategies. I'm John Sterling, editor-in-chief of Gen, and I'm going to serve as the moderator. After the panelists make their presentations, there will be a question and answer segment. Feel free to send a question for our panelists at any time during the webinar. Type your question into the Ask a Question box on the lower left of your console, and then hit Submit. The panel will try to answer as many questions as possible during that time period. So if everyone's ready, let's get going. Dr. David Gallus will be our lead-off presenter, so I'm turning the program over to him. David?
Thank you, John. As you all know, changes in biological research are accelerating rapidly, and technology is having a major impact. And we're here today to talk about next-gen sequencing and the impact it's having on biological and medical research and practice. And there has been a major revolution in technology over the last couple of decades. And what I'm going to do in this talk is first give you a little bit of history, talk about some of the major technical advances that it's enabled, discuss the challenges, give you a little bit of a view about where next-gen sequencing is going, and then draw some conclusions. So you've all probably seen this slide. This is a view of the time profile of the cost of sequencing, or upside down it would be would reflect the speed of sequencing, and the upper line is the famous Moore's law, twofold change every 1.5 years. That has reflected the development of the computer chip technology over the past 70 years or more. Sequencing followed this profile until about 2008, when a major technical shift took place and a dramatic break occurred. At that point, it began changing almost tenfold over every 2 years. And the last couple of red dots indicate where this technology is now. And the question is, how did we get here? Well, let me give you a very brief bit of history. Right. So first of all, the real revolution took place in the 70s, when it went from simple biochemical methods to find one base at a time to actually being able to read full reads of fragments. And then in a set of developments that won a Nobel Prize for Fred Sanger and Wally Gilbert, the Sanger and Maxim Gilbert methods finally relied on electrophoresis to sequence entire fragments, radio-labeled fragments. Sanger sequencing, which was based on synthesis, came to dominate and because of its speed and ease of use and its adaptability to automation. And so the next step after radioactive fragment sequencing was to add fluorescence, and that enabled the development initially of the ABI sequencer and several others at that time. The next development was the change in the electrophoresis, which occurred during the 80s. The development of that technology to capillary electrophoresis enabled much longer reads, faster information acquisition, and so on. And there were some other technologies, but we will not discuss those any further. Now the next change in technology is really the next-gen, what's called the next-gen sequencing technology. Began with high-level parallelization of the technology, and that was done largely by two companies, Solexa and Lynx in the U.S. and the U.K. And the essence of that technology is the development of individual DNA clones or clusters on slides, and the parallelization that provided, sequencing by synthesis again, but one base at a time, and then imaging the spots, the colors indicating which base was added. That sets of cycles, although it sounds a bit awkward because there were millions of spots on a slide, really rapidly increased the data acquisition. Now, what's happened over the past number of years is the development of this into machines, primarily by Illumina, but several other groups and companies as well. And these clusters of DNA have been imaged more densely, and the increase in the rapidity of the imaging, and finally the ability to analyze these images and acquire data has been the next major change in sequencing technology, and that's what we call next-gen sequencing. So once reads are obtained from these spots, putting it together into assembling it into information about longer DNA sequences or RNA sequences needs to be done. And there's basically two different principal kinds of reads, and those are either a simple single read or what's called paired end reads, which is reading two ends of a fragment of DNA that are a known distance apart. And that has several advantages over the single end read because it allows more accuracy in mapping the reads, putting them together, and much better detection of genome rearrangements and other aspects that we'll discuss in a moment. Obviously, this all requires much better quality informatics tools and computational methods in order to allow this to take place. And so the technical advances that are enabled by next-gen sequencing are really many. Primarily the first one, of course, is the individual genome sequences can be human sequences can be obtained, either individual cell clusters like tumor sequencing or just the genome sequence of an individual, and we'll talk about that in a moment. In addition, the ability to do RNA sequencing, that is, of the content of the RNA content of cells, is also changing the way in which that kind of data is used. That enables by read counting, enables the expression levels to be obtained, enables the determination of which exons are used, so alternative splicing, and even RNA editing by comparison with the DNA genome. In addition to that, messenger RNA reads, a lot of non-coding RNA, which have become increasingly important in biology, are being obtained by these methods. In addition, we won't talk about this, but epigenetic marks on the genome can also be read routinely. And finally, chromatin structure can be obtained using a next-gen sequencing technology by looking at three-dimensional structure, by doing cross-linking in a technique called Hi-C, and ChIP-seq, that is chromatin immunoprecipitation sequencing, is used to obtain information about where histones are and where transcription factors and other chromatin structural components are located. Well, let me give you a quick example based on some work we did a few years ago where we sequenced the first family of 4, and that was a sequence of 2 unaffected parents and 2 children that had both had 2 different genetic diseases. The Miller syndrome, which is a limb malformation, and a lung disease. This was done not with the Illumina sequencing, but with complete genomics technology, which I won't talk about, but the principle is the same. So what was done here was that the sequence of all 4 individuals, as indicated in this slide, in the upper left-hand side, you can see the color coding of indicating the chromosomes of mom, dad, and the 2 kids. Basically, if you look at the 2 children, any segment of a chromosome can fall into one of 4 states. Either they can be identical, that is, they got the same chromosome from mom and the same chromosome from dad, or they can be non-identical, they got neither the same, or they can have the same chromosome from mom and the different one from dad, or the same from dad and a different one from mom. So that's indicated in these 4 different codings, color codings. And if you look then on the bottom of the diagram on the right-hand side, what's indicated there are the scores for these individual types of states of the chromosome based on the variants that were scored. That is that allows one to distinguish the chromosomes from mom and dad and so forth. And the color coding along there is quite clear. You see in that bottom panel. And everywhere it changes color means there's been a recombination that's taken place, which is encoded in the diagram of chromosome 4 shown on the bottom as an example. Well, now if you look at the using this information to try to find the genes, it's indicated in this slide, several assumptions were made here. One is that that is a very good assumption, is that the defects are recessive. That is, they require 2 defective chromosomes, one from mom and one from dad. And as you can see on the left-hand side, there's 2 ways in which this can occur, and because of the rarity of these diseases, we assume that it's going to be a compound heterozygote, that is a different mutation in mom and a different mutation in dad. This can be seen from the recessive nature. It can be seen by just looking at the fact that neither parent has the disease, but both children do. The other assumptions are that because both children have both diseases, that the genes that determine these will reside in regions that are identical in both children. So potentially the twin regions. The fact that it's very rare means that they're not going to be common variants, not common SNPs, but rather very rare variants that occurred in the mom and dad and came together in the kids. And finally, the other assumption that was initially made was that it was going to be in a coding region, which is the simplest assumption to make. Well, when one makes these four assumptions and applies them to the genetic data, the amazing thing that emerged was that there's only 4 candidate genes out of the more than 20,000 genes that actually fulfill these criteria. And looking in the next slide, you see the chromosomes for the kids, the genomes for the kids, indicated in a slightly different way, using the coding where the left half of the chromosomes are the state for the father and from the father, the paternal state, and the right half is the chromosome from the mother, and you can see all the recombination points. And what's indicated in these twin regions are the 4 candidate genes. Well, to make a long story short, looking at the alternative or other patients, when we were able to identify the Miller's gene as being the one on chromosome 16 is indicated, and the ciliary dyskinesis gene being identified as the one on chromosome 5. And so this is an indication of one major example of the kinds of things that can be done with next-gen sequencing. Well, what are the difficulties? What are the problems with next-gen sequencing interpretation right now? Well, let me just point out to a couple. The first one is, of course, genetic phase determination. And what we mean by genetic phase determination is the variants. If one has 2 variants, the question is, are they on the same chromosome or are they on different chromosomes? And that information is not generally available directly from sequence, but it is inferable when you have full family sequence. But if you only have one individual, then it's only using very long reads and making various inferences about the genotypes that you can indicate probability of what the phases are. Long reads will continue to contribute to this, and the longer the reads get, the better off we'll be in determining phase. Similarly with rearrangements. Now another a problem which has not been identified commonly, which is worth pointing out, is in the human genome there are hundreds of regions where there are tandem repeats. And if one has variants in 2 different tandem repeats next to one another, it is very difficult to tell in principle whether they're in different positions in the chromosome or whether they're heterozygotes. One indication might be that, as shown in the diagram on the right-hand side with the blue looking like a histogram, this is a diagram of the coverage for one of the regions. I think this is chromosome 4, in fact, and where you see the elevation, the doubling of the coverage, that's an indication that what's on the reference genome as a single repeat is actually, or as a single region, is actually a repeated region, in this case of something like 40,000 bases. And so these so-called compression regions will be important for doing genetics either from tumors or overall inherited genomes. And the better we understand the details of the human genome, the reference genome, and the better the bioinformatics tools are, the easier this will be. There are other problems that I'll quickly mention in RNA analysis, in RNA sequencing. Alternative splicing is still very hard to measure accurately for a number of reasons. Short RNA is hard to measure quantitatively by RNA-seq because of biases in both the RNA purification steps and library preparations. And of course, improvements in informatics are constantly needed and are coming along. In addition, there's RNA outside cells, most notably, although not quantitatively, primarily in microvesicles like exosomes. And I would just mention in passing, anyone interested in that, the NIH has an extracellular RNA consortium program that has recently been started. So where we're going next is that the present technology is certainly getting better, cheaper, faster, more reliable, and the way in which that's occurring is that the density is increasing, the rapidity of the cycle is improving, and the accuracy of the reads is improving. The next technology that's probably going to revolutionize things is single molecule sequencing. And there have been a number of attempts. PacBio technology is one that we're waiting to see the outcome of, as it has a number of machines that are already being used, and accuracy is the major problem there as far as we know. The next major change is going to be nanopore methods, and if you want to read about those, the science article indicated at the bottom of this slide is a good place to look. The advantages of this will be that the cost should continue to drop, and that we are going to be increasing the number of bases per unit time that we can read. Finally, the conclusions then are that this technology is enormously powerful. It's changed dramatically, and increasingly it's biology, not technology, that's going to be the issue. Sequence information, as we learn more about the biology, is going to become much more of a use in medical practice, and it will be informative and cost-effective in the future. And the speed will change increasingly, or will increase in the next couple of years, I predict, so that we can expect major shifts to continue to improve. Thank you very much. Back to you, John.
David, thank you for that excellent introduction to next-gen sequencing and for providing your insights on where this technology might be applied in the clinic in the future. If you're just now joining our webinar, welcome. As I mentioned earlier, we'll be conducting a Q&A segment following the panelists' presentations. Please type your question for all of our panelists into the Ask a Question box on the lower left of your console and then hit Submit. Our second panelist, Dr. David Smith, is ready to begin his presentation. David?
The clinical uses of next-generation sequencing at the Mayo Clinic include a variety of different things. It starts with small gene panels, and I'm going to give the example of the very first one that went live, which is the hereditary colorectal cancer gene panel. There's also plans in development for a more broad cancer gene panel. And then other uses include transcriptional sequencing, or RNA-seq, made pair sequencing, which is a very powerful technique to look for rearrangements, copy number variation, and as a potential replacement for array CGH, and finally, whole genome sequencing. We have a clinical genome sequencing laboratory, the CGSL, which is a clinical shared services laboratory. It's named the Clinical Genome Sequencing Laboratory, and it's involved in clinical production and new test development, but it does not own any of the tests which it develops. In terms of next-generation sequencing at the Mayo Clinic, this is actually housed in two different areas. The first is the Mayo Medical Genome Facility, which is separate from the clinical sequencing facility, and then the other is the Clinical Sequencing Laboratory. There was $2 million invested in 2012 and 2013 so that we were able to have an old Illumina Genome Analyzer 2X 3 MiSeq, Illumina MiSeqs, 2 Illumina HiSeq 2500s, and a Life Technologies Ion Torrent instrument. The bioinformatics is a very important part of this laboratory, so that this is involved with the Department of Laboratory Medicine and Pathology. They do routine data analysis on clinical samples, high-performance computing cluster, a next-generation sequencing workbench, and also a target panel development. And there is a series of 5 different tests which are under development across different divisions and laboratories in DLNP, all using small gene panels. In terms of the hardware, this is a 2-phase thing. In the very first phase, which is up currently, it's 3 64-bit, 8 CPUs, 32 gigabytes of RAM, 2 64-bit with 2 CPUs with 4 gigabytes of RAM. But then in terms of our phase 2, this is going to be extending that a lot in terms of having 7 64-bit, 12 CPUs, 2 64-bits with isolon storage for all the data which is going to be generated, which will need to be stored. In terms of the clinical next-generation sequence, the overall program in review is summarized in the pictorials that are seen here, everything from the top left, which is sample collection and preparation, to the next-generation sequencing, the data storage, the compute and analysis function, a consultant workbench, and then the generation of an interpretive report so that it'll be easy and transparent to return the results to the patient. In terms of the instrument considerations, there's a variety of the next-gen sequencing platforms that are currently available, and at the time when the clinical lab was trying to decide, they were looking between these various options. Many of these, of course, are from Illumina, but you also have an Ion Torrent and a Roche machine over here. And one of the issues and questions is whether we felt we would need to do cross-validation, so data would be generated on one next-generation sequencing platform and potentially validated on another. In terms of informatics, hardware and software, a lot of different options were considered in terms of what we should develop ourselves and what we should be getting outside. So in terms of hardware, data storage, case exploration, novel discovery, versus the software like alignment, variant detection, and these were some of the other things which were explored in the overall process as this was begun to be set up. This actually shows the entire workflow of all of the aspects of our very first small gene panel, which I'll describe in more detail in a couple of slides. This is the hereditary colorectal cancer panel, and this shows all the various processes and workflows from getting the sample, make sure you have sample ID, the next-gen sequencing component, to the data analysis, to the interpretative workbench, and to taking the final report to the patient. In terms of the clinical next-generation sequencing, they had to develop a very nice system for data visualization and decision support, and they developed a consultant workbench. And its function is to transform annotated data into an electronic result worksheet of the highest quality as quickly as possible. And this was set up. And then the enhanced reporting is basically, especially since you're dealing with multi-gene panels, it's a new way to report the data back to the physician, and that the patient has to be considered in this new and more complex world. This is just an example of one potential type of report on a Mayo Clinic paper for a specific alteration, and then the next slide actually shows what you would actually see. So that if the patient was screened and one of the genes happened to be APC that had a mutation, this would be the patient showing the APC gene, but then some information that could be provided both to the clinician and to the patient about the specific gene that was actually being mutated or was mutated in that patient. In terms of developing this, the very first test that was used, again, was the hereditary colorectal cancer panel. And they were interested in expanding the current 4-gene panel. These were 4 genes which were analyzed by Sanger sequencing, 1 gene at a time, to a test menu of 14 for more comprehensive testing. So this is taking all 14 genes that are known to or suspected to have a role in hereditary colorectal cancer. And basically, it was also a pilot that Mayo could utilize for the development of a small gene panel. And once this was in place, it would be relatively straightforward to substitute other genes so that you could have panels, for example, of cardiovascular disease, neurodegeneration, etc. Now, in addition, because sequencing by itself does not see all things, certainly changes in copy number, there was also a plan, and it is in place, to do concurrent array CGH to determine if there has been changes in copy number. So the very first clinical test, they wanted to develop this panel. They wanted to have 100% coverage with a minimum sequence of 100x at every single nucleotide that was being queried. Then the issue in question as to whether or not Sanger sequencing would be needed to verify if you were either of the mutations that were detected or if you could not see every single nucleotide. The overall thing that was selected was Agilent SureSelect to pull down the targets. This was originally done on the Illumina GA2X, but it's migrated subsequently to the MiSeqs. In terms of storage, this is Isilon bulk data and the development of the Mayo workbench and enhanced reporting, all part of the clinical test. And once this is now live, Mayo is now discussing and developing additional gene panels. One of the other things that is currently under development, but it's not clinically live yet, is a pan-cancer gene panel. And this is a panel consisting of 450 cancer-related genes. The genes will be brought down by SureSelect again, and then Illumina and ion Turing cross-platform validation to see which alterations can be detected. In addition, there's work in progress to develop transcriptome sequencing, or RNA-seq, because of the importance in determining gene expression. A very good example is the fact that if you're studying oropharyngeal cancer, a cancer that I happen to study, it turns out to be extremely important to know what is the actual expression of the oncoproteins E6 and E7, and this is something you don't get from DNA sequencing, but can get from RNA-seq. It also gives you a comprehensive list of gene expression, the ability to detect novel fused transcripts, and it raises the question, could this too be a normal, routine part of cancer care? This actually shows the workflow that has been developed by the bioinformatics team at Mayo for analyzing RNA-seq data. So data is obtained at the top, and these are the various ways and that this can be analyzed so that the report can come back with changes in gene expression, can come back with single nucleotide variants which have been detected in the transcripts, and can also come back with potential novel fusion transcripts which were present. A very powerful technique that has been worked on a lot, especially by the biomarker discovery program within Mayo Clinic Center for Individualized Medicine, is the power of mate pair sequencing. And this is a powerful technology that analyzes genomes by breaking the genomes into larger pieces of 3 to 5 kilobases in length, circularize those pieces together in such a way that you could just enrich for the 2 end pieces, which were initially 3 to 5 kilobases apart. This is actually illustrated on this shown right here, where the DNA is isolated and fragmented. You circularize the DNA by putting biotin at the 2 ends, and then when that DNA is broken into small pieces, the only fragment that is saved and that will stick to a magnetic bead coated with streptavidin are the 2 end pieces, which were initially at some distance apart. And when those are sequenced by paired end sequencing, one generates sequence which would be aligned on the genome at a position of about 5 kilobases apart, unless there's been an alteration in that region. An insertion would make things too far apart. A deletion would bring things too close together. Now, one of the most powerful things that this was developed by George Basmantis, who heads the biomarker discovery program, was his binary indexing algorithm, which is a very powerful algorithm that you could use to simplify the data analysis of the mate pair sequencing, and with this, to identify novel alterations. And here, for example, is two pieces of DNA, one 5 kilobases apart, and one over here indicating 2 pieces that normally would not be suspected to be next to each other. This could be a piece from one chromosome, and then green could be a piece from another chromosome, in which case this would be an intrachromosomal translocation. But this could also be a large deletion that now brings 2 pieces from the same chromosome much too close together. And using this powerful technique, this is actually from one of the publications that came out of this work, which is the detection of a very novel ALK translocation in the lymphoma. And here you see all of the mate pairs that go across between chromosome 7 and chromosome 6 that located specifically this balanced reciprocal translocation and the 2 genes that were involved in that. Another very powerful application is from some work that we recently published, and this shows actually the site of a human papillomavirus integration in an oropharyngeal cancer, and here is the positions on chromosome 1, which is the chromosome where the integration occurred, and these on this side is the pieces of the human papillomavirus 16 that integrated into that spot. And mate pair sequencing is very powerful for the identification of both of these types of alterations, and this has a number of clinical uses. So, for example, one of the clinical uses for mate pair sequencing is as a replacement for array CGH. Because you're sequencing 100 base pairs at the end of fragments that were originally 5 kilobases in length, it's only necessary to do significantly less sequencing to get a high-resolution analysis across the genome, and this may be a very cost-effective way to get a very powerful replacement. It also could be used, as I showed on the previous slide, to look for viral integrations in cancer. And finally, the identification of novel translocations could itself be a very powerful tool for monitoring cancer in terms of how effective therapies are and when that cancer is coming back, especially if that's combined with the power of digital PCR. Whole genome sequencing is not yet done for anything clinical at Mayo, but it is being done as part of one of the research projects, which is a beauty project. And this is a project that is working with breast cancer and trying to figure out the specific alterations of breast cancer, and it's tying this together with mouse avatars, which are generated by making xenografts from those cancers and trying different drugs on avatars to see if they're effective. The only thing here is that you might do 100 gigabases of DNA sequencing for a normal genome, and you probably want to do in excess of 200 plus gigabases for a cancer genome, depending on how heterogeneous you think it might be. This is actually the pipeline that was also developed by our bioinformatics department. This is Jean-Pierre Cochet and his group for the analysis both of exome sequencing data and genome sequencing data, and these are the various pipelines that are followed by these in order to take that data, to align it, and to determine what is going on with the data that you're looking at. This is the acknowledgments of the people that were all intimately involved in this, from Matt Ferber, who heads the clinical lab in next-generation sequencing, George Vasmontes, who heads our biomarker discovery program in our Center for Individualized Medicine, Jean-Pierre Cochet, who heads bioinformatics, and then some of the work of some of the scientists who were instrumental in this work. Thanks.
Thank you, David, for demonstrating to our audience how next-gen sequencing is used clinically at the Mayo Clinic. That's why we thought this webinar would be appropriate right now because of the technique's increasing clinical utility. Before our third speaker begins her presentation, I'd like to remind the audience of our Q&A segment that will follow her talk. Please type your question for any or all of our panelists into the Ask a Question box on the lower left of your console, and then hit Submit. Dr. Elaine Martis is now ready to talk about the use of cancer genomics to guide therapy. Elaine, the floor is yours.
Thanks very much for the invitation to present this material today, and I'm happy to be able to talk about how we're using cancer genomics to guide therapy for cancer patients. On my first slide, I show a picture of Dr. Janet Rowley, whose pioneering work in the 1970s using microscopy to study leukemia chromosomes really began to firmly establish that when we saw aberrations in cancer, these were directly related to aberrations in chromosomes. And this firmly established an idea that was advanced by Theodore Boveri in the 1900s, that changes in chromosomes actually were causative in terms of the development of cancer. If you look at the next slide, you'll see that the chromosomes that Dr. Rowley originally was studying under the microscope were further elucidated by a study that we published in 2011. This was a study of a patient who was thought to have the a type of leukemia called acute promyelocytic leukemia, studied by Dr. Rowley, where she identified the translocation between chromosomes 15 and 17 originally in these patients that she evaluated under the microscope. Nowadays, our microscope is a much more high-resolution scope, the next-generation sequencer, that allows us to see at nucleotide resolution the changes that go on in the chromosomes of cancer patients. In this patient that we described in the JAMA article, thought to have the type of leukemia caused by the canonical 1517 translocation, we discovered that while this translocation couldn't be seen by conventional means under the microscope, our reassembly of the patient's chromosomes showed that rather than a translocation having taken place, a large insertion of chromosome 15 into chromosome 17 had occurred. But the net result was exactly the same as is often seen in the translocation of acute promyelocytic leukemia, namely two genes, PML and RAR alpha, shown to the lower right-hand corner of this slide, had been fused together by virtue of the insertion, forming a fusion gene that's known to be necessary and sufficient for patients to develop this disease. On the next slide, we show a study that we published in the early 2000s, just coming out of our work and others on the International Human Genome Research, the Human Genome Project. These approaches preceded the advent of next-generation sequencing, and so used conventional PCR and capillary sequencing to evaluate several genes in the genomes of lung cancer patients that had been shown to either be resistant to or to have a dramatic response to a new class of drugs at that time known as tyrosine kinase inhibitors. On the slide, we indicate the area blown up of the tyrosine kinase domain of epidermal growth factor receptor. And in what we found in this study was that patients who had a dramatic response to this tyrosine kinase inhibitor called ERESA were found to have mutations in the tyrosine kinase domain, and these are indicated in the large blowout section of the tyrosine kinase domain for this gene. If you move to the next slide, you can see that in just 10 short years, the knowledge about aberrations in lung cancer has increased dramatically, including the EGFR alterations that are shown in the green slice on the pie graph. We now know of a plethora of other types of genes that are altered commonly in lung cancer, and this was reviewed by Will Powell and Hutchinson in the Nature Medicine review that's indicated at the bottom of the slide. However, it's still the case that a very large slice of this pie remains unknown, indicating that more studies need to be done to evaluate the genomes of lung adenocarcinoma patients. However, in 10 short years since that first description in PNAS by our group in collaboration with Dr. Powell and Dr. Harold Varmus, when one is currently, nowadays, diagnosed with a lung cancer, a biopsy needle can be inserted, as shown in the upper right-hand panel, and a small portion of tumor cells can be removed for genetic analysis or evaluation. One might find, for example, the same EGFR mutations, which would be indicating that the patient is likely to respond to one of the listed tyrosine kinase inhibitors. Alternatively, if you have both EGFR and KRAS mutations, we know that tyrosine kinase inhibitors will not work to help control and diminish the disease. And so these patients go to the standard of care of surgery, radiation, and chemotherapy. More recently, the ALK fusion genes, the genes that fuse with ALK, that is, have been shown to be responsive to a new drug called crizotinib. And so when these ALK fusions are detected, the patient can go directly onto the crizotinib drug. And in many cases, both with the tyrosine kinase inhibitors as well as with drugs like crizotinib, these patients achieve typically a very dramatic reduction of their tumor burden in these circumstances. And so this is really showing over a short 10-year period how genomic medicine has had an impact for lung adenocarcinoma patients. Now what remains is for this to be more broad-based across multiple types of cancer. In the next slide, we show the study that we published in Nature in 2012 looking at estrogen receptor-positive breast cancer, where we sequenced through 47 patients by whole genome methods and cataloged the genes that were frequently mutated. What we showed in this study published in Nature was that MAP3K1, highlighted at the upper left, was a gene that was frequently mutated in estrogen receptor-positive breast cancer, And this was the first description of this gene as frequently mutated. Later studies by other groups studying the basal subtype of breast cancer show that this gene is never mutated in basal type cancer of the breast. And so our finding here looks like a unique mutation of this gene in estrogen receptor positive disease. As interesting as this novel finding was, you see the additional genes that are highlighted, RUNX1, MyH9, MLL3, et cetera, below on the lower left. And the excitement around these genes being identified as frequently mutated in breast cancer is that these were commonly thought to be genes that were mutated only in blood cancers or leukemias. So this was also a unique finding that these would be frequently mutated in estrogen receptor positive breast cancers. If you move to the next slide, what we can see is that, however, unlike lung cancer where there are multiple druggable targets, this is really not the case so far in estrogen receptor positive disease. However, in the same study in Nature, shown in this table, what we published was the fact that we had identified multiple other genes that didn't meet the criteria for significantly mutated, as I showed in the previous table, but nonetheless were found in a number of patients, even with an admittedly small study that we had. And all of these genes, as you can see in the panel identified as matching drugs, have drugs that are actually FDA approved or planned for use with these types of gene alterations that we were able to identify in breast cancer. However, of the drugs that are available for these drug-gene interactions, you'll see that only the PI kinase 3CA drugs are being tested in breast cancer at this point in time. And so this points out another difficulty with regard to applying cancer genomics to help identify therapies, namely that our paradigm for clinical trials evaluations of drugs that correspond to specific altered proteins in cancer needs to change so that we can test these drugs in multiple tumor types at the same time and get FDA approval when there are appropriate responses that are identified. In the next slide, you can see the first group to really apply the overarching goals of cancer genomics to guide therapy was published in 2010 in Genome Biology. In this report, the group identified on the author list, largely from the British Columbia Cancer Center and Genome Sequencing Center, were able to successfully extend the life of a patient who originally presented with adenocarcinoma of the tongue and subsequent metastases by applying the principles of cancer genomics, identifying key mutations that look like they would be responsive to specific targeted drugs, and getting those drugs to that patient. And through a series of this cycle where they tested the genomics of emerging metastases in the patient, identified new drugs, and were able to get those drugs to the patient, the life of the patient was extended. And the conclusion from this study, which I think really sets the stage for this paradigm in current clinical medicine, is that we have to be able to do genomic comparisons as a routine part of cancer pathology to help guide therapeutic options. But on the next slide, I pose the question, is this confluence of next-generation sequencing, our abilities in cancer genomics, as I've illustrated so far, and the application of targeted therapy the perfect storm? Or on the following slide, is this just a pipe dream, where the pipe dream is sort of a phrase that's often used. It means a hope, wish, or a dream that is impossible to achieve or not practical. And the source of this is actually an allusion to the dream that were experienced by smokers of opium pipes, and that's the origin of this word. And so I think this is really sort of illustrative of the multiple complications that are being experienced in terms of really bringing the promise of cancer genomics and therapeutic intervention to reality in our current clinical setting. At our place, as shown on the next slide, we're approaching this from a very comprehensive standpoint. Rather than just choosing a small select list of genes that we think might be involved in cancer and that might be druggable, we're instead going for a very comprehensive and integrated approach that starts with whole genome sequencing to yield the full variety and types of variants that we know are causative for cancer, everything from point mutations to amplifications or deletions of genes to large structural variations, such as these translocations that Dr. Rowley originally described by looking through her microscope. In addition to the whole genome data, we also are producing exome sequencing data. This is the capture methodology to just select out the 1.5% of the 3 billion base pair human genome that encodes the known genes in the genome. And in exome sequencing, we obtain a number of key value-added points. First of all, the separate calling of exome data, tumor versus normal, will validate any of the whole genome discoveries in terms of the single nucleotide variants and indels that have been identified there. And this gives us confidence that we're getting the right answers in terms of the mutational landscape for each patient. In addition, we are able to integrate the whole genome sequencing data with the exome data for tumor normal, and this gives us a net higher coverage depth that allows us to evaluate the clonality of different mutations. Are they low-lying variants that may rise to prevalence under some sort of negative or positive selection and come back with a different clone of the disease in the metastatic setting, and so on and so forth. So this really gives us the ability to look for low-lying variants, and in particular to pay attention to those that are druggable or already known to lead to acquired resistance to targeted therapies. And then lastly, we're also looking at the RNA that's expressed by the tumor. Here we can really mine out information that we will never see by just sequencing DNA alone. For example, genes that are overexpressed relative to the normal tissue. In particular, these may be caused by changes in methylation status or histone binding that we simply can't detect by just sequencing native DNA. We also identify those point mutations that were identified initially from whole genome or exome data that are really being expressed in the RNA. And in tumor types, especially those with a very high mutational burden, the number of mutations that are actually expressed as RNA is surprisingly small. Typically around 40 to 45% of those mutations are actually expressed. And then lastly, any gene fusions that we're detecting from our structural variant analysis in the whole genome can also be identified from RNA-seq. And again, this interplay of DNA and RNA sequencing data gives us confidence that our calls are correct and not simply false positive results. In the next slide, what we have to do once we've cataloged through all of these variants is really to apply the known annotation on the human genome. And this pie graph points out how much we know about the human genome. We understand the exome, which I talked about earlier. Those are the known genes in the genome. We're beginning to better understand the approximately 9% that's highly conserved across species through evolution, as well as those that have been illustrated to be regulatory, so controlling when genes are on or off. And there's about 42% that we really don't understand, but is highly unique sequence data. So we're cataloging mutations there by whole genome methods in the hopes that over time we'll better understand those regions that are frequently mutated. But annotation is really important, as now illustrated in the lower right-hand corner of the slide, because it really tells us what we're looking at above and beyond A's, C's, G's, and T's. This illustration is from another paper that we published in 2011, a companion to that JAMA paper that I mentioned earlier in the talk, where we really showed that this patient had missed out on a Lee-Fraumeni TP53 novel mutation. In particular, this was detected as shown in the upper panel because in the skin, which is the comparator normal here, this patient had therapy-related AML, so her skin is taken as the normal because the bone marrow and the blood contain the tumor. you can see the marked region of heterozygous deletion where we just got lower coverage, but this is remarkable in the bone marrow, the tumor in this case, because it now goes to complete homozygous deletion. So this is just phenomenological until you apply the annotation of the human genome, and you can see in the lower panel that the region of deletion is in the gene TP53, a very well-known tumor suppressor, eliminating exons 7, 8, and 9, and this would have never been detected by conventional sequencing because these exons actually exist in one copy of the patient's 2 chromosomes in the normal genome. And so you would always get products for those, and they would always sequence like they were supposed to from PCR and capillary sequencing. It's only with next-generation sequencing that we have the ability to detect this. And so lastly, what we do with all this information is we comprise a Circos plot, which is shown on the next slide, really a Cliff Notes version of every patient's genome, where you can see the colored blocks around the outside of the circle reflect the different chromosomes numbered from 1 through X and Y. The words that are written to the outside of the circle are indicative of the genes that have single nucleotide variants or insertion deletions. The gray area in the center is indicative of copy number changes, and then the colored arcs and spikes in the center of the circle are representative of translocations, inversions, deletions, and other types of structural variants that we identify. And literally for every patient that we sequence, the Circos plot looks highly different. The genome finds a multitude of ways to put together different causative variants that lead to the development of cancer in each and every patient. So on the next slide, there's a very complex series of steps that we take to now try to make a therapeutic interpretation from this very complex and comprehensive look at each patient's genome. Shown on the right-hand side are all of the different types of variations that we can identify through the analysis that I just described. And importantly now, to clinically prioritize these variants, we have to do a couple of important steps. First of all, shown in the light green box in the center, we have to functionally annotate every gene that's altered in terms of the protein and what impact that mutation might have on its function. And secondarily, and very importantly, of those functionally altered genes that we're predicting, we have to identify which are going to be activating, because we know that, as shown in the third block, cancer pathways are what are impacted, not just individual genes or their protein products. Namely, what we have to do is take the candidate genes that are functionally altered and activating, layer those onto the known cellular pathways in cancer, and then use information shown on the right-hand side from the drug-gene interaction database to really decide where best to drug that pathway for the altered activated genes that is in it. And the drug-gene interaction database is really a clearinghouse for information, because there are a lot of sources of information about genes that are mutated and the drugs that interact with them. And so this resource is publicly available through the internet and really would allow a quite simple lookup of gene-drug interactions, list those out, and then provide information about where to find more information about those drugs, the clinical trials that are available that are testing those drugs, and so on and so forth. All of this information then distills down to what we refer to as clinically actionable events, also known commonly in the parlance of cancer therapeutics as the report, which is really going to be what we hand to the patient's oncologist that's had this type of analysis, so that he or she can then go off and consider whether any of this information is relevant to the patient's care. The next slide just shows a live illustration of the drug-gene interaction database with the URL listed down at the bottom of the slide in the lower left-hand corner. The search interactions page is shown on the left-hand side of the slide and really is a very simple interface into which the gene names of interest are typed. Several parameters down below can be set, including whether one wants to see only anti-neoplastic drugs displayed, and then a click on the green button that says Find Drug Interactions advances to the next slide, which shows the interaction search results coming out of this particular query of DGIDB. And just for illustration, you can see the ABL1 kinase has been identified as one of the genes of interest. There are numerous drugs available for this gene when it's altered. In cancer, these are all inhibitors as shown in the display, and if we wanted to get in more information about any or all of these drugs, a simple click on the green source button, in this case for My Cancer Genome, would link out the user out to the page for My Cancer Genome that contains more information about this particular drug, any published references, etc. So My Cancer Genome is a well-curated database of drug-gene interactions and would just provide the user with much more information than we could ever coalesce. And this is the case for all of our source databases providing drug-gene interactions. The next slide just shows an illustration of what DGIDB can provide for a number of patients. These are lung cancer patients that we published a paper in Cell in 2012, as referenced in the lower right-hand corner, where you can see that in this very colorful diagram, we're looking at gene targets, a class of therapy that corresponds to those. They're listed on the right-hand or left-hand side of the slide. On the right-hand side is the targeted therapy that was identified in each case. And this is just a collective group of smokers and never smokers for which we sequenced whole genome as well as RNA-seq data and then identified drugs that would be applicable. I think the most important thing about this slide is really just to illustrate how many drug target hits we identify. And in particular, smokers have a very high mutational burden. So you can see that there are lots of different colored blocks corresponding to lots of drug-gene interactions. But I would also point out that for never smokers who typically have a paucity of mutations, many of which are not druggable, we've identified a significant number of hits by sequencing RNA. Those are the green blocks that are identified as overexpressed genes. And so this just illustrates the value of sequencing RNA, as I was talking about earlier, above and beyond simply sequencing DNA alone. Then the next slide is really just another illustration of what one can identify. This is, again, the breast cancer example, hearkening back to that previous table that I mentioned from the Nature 2012 paper on breast cancer out of our laboratory. And the reason why we've done this, and this is an illustration from the Nature Methods paper describing DGIDB, is for the reason that I mentioned earlier. Very few FDA-approved targeted therapies for breast cancer. We took the compendium of 315 recurrently mutated genes that have been identified across a multitude of breast cancer studies already published, and applied these into DGIDB, as I just illustrated, to identify numerous potentially druggable genes from 6 sources. This reinforces that small, focused look that we published in the Nature paper in 2012. The lower graph is really indicating the number of these genes in potentially druggable categories versus the number of genes that are actually targeted by a known drug. And you can see in this regard that pharmaceutical development has a long way to go towards identifying drugs that will correspond to many of these targets. The next slide is really just a list, if you will, of many of the other challenges that remain, really bringing cancer genomics in as part of the therapeutic equation for most patients. And I won't describe any of these in detail, but people who are trying to do this at different institutions can probably relate to this list of topics. At the bottom, in the next slide, one of the most pressing issues that faces us today in the context of targeted therapy, however, is this notion of acquired resistance that I referred to earlier. Namely, what we see is that patients that have a drug-gene interaction indicated by genomic testing or even single-gene testing can get that drug, they can take it and often acquire a dramatic response and reduction of their tumor burden. But many, if not all of these patients, represent with advanced metastatic disease, which is known as acquired resistance. So their tumor cells find a way, if you will, to invent around the blockade that's been put on their genome by the targeted therapy. And if you go to the next slide, what you can see is now a very recent report from a group looking at combined targeted therapy, where you're drugging 2 points in the pathway, one just downstream of the other. This was thought to be an approach that might get around acquired resistance from monotherapy, is just to give the patient 2 therapeutics. This notion came out of studying acquired resistance and the nature of acquired resistance. However, in this report that came out at the end of last year, in patients with melanoma who were getting 2 drugs, one to attack the BRAF V600E mutation or V600K that's very prevalent in melanoma, about 50%, as well as the downstream MEK protein, also were illustrating acquired resistance on this combined therapy. So we still have a long ways to go to make this really work out to be something that's going to be giving patients what is known commonly as a durable response. And one answer to this maybe is shown on the next slide, which is the use of immunotherapy. This was described in Science last year as the breakthrough of the year. We published a paper in Nature 2012 looking at the type of genomic interactions with the immune system that lead to tolerance of the cancer, so-called immunosuppression. And in this approach, we're now moving forward to identify, using genomics, the specific alterations in the tumor genome that produce highly unique proteins or peptides carrying those mutated amino acids that present the most strong stimulus to each patient's immune system. So as shown in the slide, we biopsy in our study metastatic melanoma lesions. We sequence the tumor and germline by exome methods, and we also sequence the RNA to find the expressed mutations that are highly expressed. We use an algorithm called NetMHC to identify the immune epitopes that are most immune stimulatory to that patient's MHC class 1 subtype. And then we use a specimen from that patient, an apheresis sample, it's called here, to really verify the algorithmically identified immunoepitopes that are going to elicit T-cell memory once they're used in a vaccine for that patient. And this allows us to refine above and beyond what the algorithm tells us. And we are anxiously pursuing this with a small FDA trial in melanoma at present, with 3 patients already having received their own their own personalized vaccine. And there are other groups around the country that are pursuing this as well. So I'll finish there with just that tantalizing thought that there may be more coming out of genomics in regard to cancer therapies. And on the last slide, I just want to acknowledge the group at the Genome Institute that produces these studies with me, as well as our investigators at our Siteman Cancer Center, who are the clinical experts that guide our work. And then listed just below them are my immunology colleagues that are pursuing the personalized vaccine approach. Thanks very much.
Thank you, Elaine, and thank you to both Davids for your marvelous presentations on next-gen sequencing in the clinic. It's really easy to see why this technology is being seriously considered or actually adapted so rapidly by clinicians because of the potential benefits it can bring to the patient population. Thank you all once again. We're almost ready to begin our Q&A session, but first I'd like to ask you to disable your pop-up blockers because a short survey on this webinar will be appearing on your screen in a moment. We'd very much appreciate your feedback on this webinar. Okay, so we're all set to begin our Q&A session, so let's take the first question from our audience. Okay, so the first question we're going to direct towards Elaine Martis. Someone asks, for patients who have their genome sequenced, does it require more experimental validation before the clinical report, or can this come directly from the sequencing data?
Hey, so that's a great question. I think historically people have considered Sanger sequencing to be a gold standard, but in many instances it's really eclipsed by the next generation sequencing approaches, which essentially are reporting information from single strands of DNA. Because this is where the library begins. So In this current iteration of next-gen sequencing moving into the clinic, it's widely accepted that because of the depth of next-gen sequencing that can be generated from single molecules, that as long as one's pipeline for analysis is reporting the appropriate metrics for coverage and quality and has been validated for that CLIA environment and process, there is no a priori need to go to a secondary or orthogonal validation strategy in advance of coalescing around the report. As I also mentioned in my talk, the interplay between DNA and RNA can be incredibly valuable in this regard to really support that the variant is not only present but is also being expressed in the RNA.
Thank you very much, Elaine, for answering it. Thank you. David Gallus, during your talk you mentioned short RNA, and the question came in, what does the speaker mean by short RNA?
Sure, that's a really important question. In the biology of the last decade or so, it's become clear that there are a lot of non-coding RNAs. Principally, first discovered were microRNAs, which are only between 20 and 23 bases long, that are really important in gene regulation. And so there's an increasing need in order to understand the biology to be able to identify the levels of these various RNAs in tissue, and actually it turns out that they're circulating in plasma as well and are increasingly being used as biomarkers. So short RNA includes microRNAs. There's a number of others which I won't go through, but representatives include short nucleolar RNAs, which are actually also present not only in the nucleolus but in plasma as well, and there are a number of others. So this is an increasingly important area, and one of the reasons that this is difficult is because in the construction of the libraries for next-gen sequencing, requires single-stranded RNA ligation, which introduce a lot of sequence-specific bias and is just now being characterized fully, and that presents a technical problem that is going to be increasingly important. So short RNAs are actually going to be increasingly important in the future.
Thank you very much, David. And David Smith, question for you. Both Illumina and Ion Torrent sell cancer gene panels in a kit. Why are you still developing your own?
That's an excellent question, too. It turns out that the Department of Laboratory Medicine and Pathology that I reside in, that is developing a lot of these tests, has wisely organized a bunch of disease-oriented groups. And these are interactive groups between basic researchers like myself and clinicians interested in specific cancers and other diseases. And the charge was to go to each of the disease-oriented groups and get from them their comprehensive list of genes that they would like to have included on the panel. And then all of these disease-oriented group panelists were then put together to do a single comprehensive panel. And then the idea is that then as we begin to utilize this panel on a larger and larger number of different cancers in Mayo patients where we have excellent clinical records, we can much more clearly see relationships between mutations and outcome, and also see how frequently mutations are across different cancers. So that's the reason that we decided to go ahead and make our own large gene panel based on the interest of the researchers and clinicians here.
Well, you answered that clearly and directly. Thank you. Well, unfortunately, we've run out of time on this webinar, but please note that the webinar will be archived for 6 months on our website, www.genangnews.com. If you missed parts of it, you can watch it again, or you can recommend it to your friends and colleagues, which we also recommend you do. I want to thank the panel again for their wonderfully outstanding presentations, and I want to say thank you to our audience for your attention and for your thoughtful questions about various topics brought up during the webinar. And thank you to Roche, whose support made this webinar possible. Bye for now.